Welcome to our webinar on gene therapy. My name is Noor, and my colleague Rulan and I have prepared this webinar as candidates for the Medical Genomics Master's Program at the University of Toronto. In this webinar, we will be answering the following questions. What are genetic diseases? How can we treat them? What are the challenges facing various forms of gene therapy? And what can we expect of gene therapy in the future? In order to understand gene therapy, we have to first understand what is meant by the word gene. This requires us to really zoom into how a person works. The human body is made up of different organs and tissues, which are in turn made up of cells. Cells work together to make organs function. There are different types of cells, and each have different jobs within the body, but they all have one thing in common: DNA. DNA is a very large molecule and is referred to as the blueprint of life because it is the instruction manual to create, grow, and reproduce life on Earth, similar to a blueprint of a house. In other words, it has all of the information that makes you, you. It is made up of 4 smaller molecules, A, T, G, and C. Each of these molecules combines to produce a unique sequence for each person, billions of letters long. The order of these letters determines your height, your eye color, your hair color, and much, much more. You have 2 complete DNA blueprints, one from your mother and one from your father. The DNA molecule contains different segments, called genes. A gene is a DNA sequence that carries instructions to make the building blocks in your body, known as proteins. To make a protein, a gene sequence is copied into a molecule called RNA, which is very similar to DNA, but more flexible in structure. The RNA sequence is then converted into an amino acid sequence, which makes a protein. Each group of 3 DNA letters codes for a particular amino acid. For example, a DNA sequence of CAG, CCC, GCC would make a protein with the amino acids glutamine, proline, and alanine. Most proteins are hundreds or thousands of amino acids long, and they have unique functions based on the amino acid sequence. Proteins can work as enzymes, which make reactions in the body go faster, or they can be structural proteins, like those in your muscles. Or they could even be antibodies and protect your body from germs, and much, much more. It is the amino acid sequence that gives proteins their unique abilities, and the amino acid sequence itself depends on the DNA sequence. The correct DNA sequence makes the correct proteins, and in turn, those proteins do their job within the body. But what happens if the DNA sequence is not correct? What if there's a different letter? Some changes in DNA are normal. It's what makes us different. But some changes can result in a genetic disease. For example, consider the word axe. If you remove the E from it, it still has the same meaning, depending on if you spell it the Canadian or the American way. Now, if the X is changed to a P, this changes the meaning of the entire word. Similarly, some changes in the sequence of DNA don't have an effect on the protein, while others can completely change it, resulting in disease. Let's go back to our earlier example. If the second A in CAG, CCC, GCC got changed to CGG, CCC, GCC, then the amino acid sequence would change from glutamine, proline, alanine to arginine, proline, alanine. Since protein function depends on its structure and sequence, this could have huge impacts on the protein, resulting in a genetic disease. Simply put, a genetic disease is a disease caused by a change in the DNA sequence of an individual, called a variant. Examples of genetic diseases are cystic fibrosis, sickle cell anemia, and Huntington's disease. To get the genetic disease, you must have the disease-causing variant. You can get this from your mom, from your dad, or even from a random mutation called a de novo variant, which can happen when your DNA is copied mistakenly. Sometimes, one variant is enough to cause the disease. Other times, 2 variants, usually one from mom and one from dad, are required to cause the disease, and you would still be healthy if you had just the one. This pattern of inheritance depends on the exact gene and disease. So say you did have a disease-causing variant that caused the genetic disease. Can it be fixed? The answer, as we will illustrate, is yes, although this can be quite challenging. The challenge in fixing most genetic diseases is that the variant causing the disease is found in every cell in your body. If you're wondering why that is, Remember that every cell in your body is made from one cell, called a zygote, which is formed when your father's sperm and mother's egg combined. Every cell in your body is derived from this, and so has the exact same DNA sequence. So if the variant was found in the zygote's DNA, then it will be found in every DNA sequence in your body. Fun fact, it's estimated that there are over 30 trillion cells in the human body. How could we possibly fix the variant in so many cells? This is where gene therapy comes in. Gene therapy is a type of treatment that involves going into your body and making changes to your genes to fix the mistakes in your DNA sequence. There are several different ways to do this. A very straightforward approach might be to replace the incorrect sequence with a correct copy. The first question that arises through this, though, is how would we deliver the sequences to the right cells? A solution to this, it turns out, is in using viruses. Typically, viruses are used because they can recognize certain cells and can actually inject their own DNA into these cells, which is what makes us sick. Now, when using viruses for gene therapy, their own DNA is removed, and so the virus becomes harmless, and its DNA is replaced with a new sequence for the desired gene that is written into it. The virus, now containing a normal copy of the faulty gene, is given to patients and allowed to infect their cells, which allows for the normal copy to be delivered to these cells. So this sounds great. Why is it not used all the time now? The problem is that this technique can have problems with specificity. The virus could randomly insert the genetic sequence anywhere in the genome, and this could cause problems if the gene is dropped in the middle of another gene, disrupting its sequence. This can cause further genetic issues, such as cancer. Another issue that arises when using viruses is that of immune reactions. While the viral particle is no longer disease-causing, it still looks like a foreign body to your immune system, and the immune system will attack it. Sometimes these reactions can spiral out of control and in very extreme cases result in organ failure. That is not to say that viral gene therapy is useless. The risk of these issues arising can be adjusted by carefully selecting and designing the delivery virus, and over the years, other techniques have emerged to deal with this. One such technique is called ex vivo gene therapy, which involves removing the patient's cells and growing them in a plate outside the body. Following this, an edited virus is again used to deliver the new sequence. The modified cells that now carry a normal copy of the gene are injected back into the patient and will go on to produce a functioning protein. This way, cells with the correct insertion could be selected and inserted back into the body, avoiding infecting the patient with the virus directly. Before using a virus, how exactly does that solve the problems we just talked about? Well, specificity of insertion can still be an issue, but in a petri dish, you can run some quick tests on the cells to make sure that the insertion hasn't disrupted any important genes, and screen for any signs of cancer. Also, the immune issue can be solved as the dish doesn't have an immune system, and we would not be directly injecting the virus into the body. However, a major limitation of this technique is that it can only be used for diseases where the cells can easily be removed from the body and then returned again. As a result, this method is usually used for disorders arising from blood cells, as they are easily accessible, like sickle cell anemia and thalassemia. In order to get around some of the issues of targeting DNA directly and targeting more diseases, RNA interference was introduced. The aim of RNA interference, or RNAi as it is more commonly known, is not to change the DNA sequence, but rather to intercept the RNA copies of the gene before they are made into protein. How does this work? In the lab, we can create RNA sequences that are complementary to the RNA produced to the cell, called silent interfering RNAs, or siRNA. A complementary sequence will then bind to the RNA that is on its way to be turned into a protein. While 2 complementary DNA strands is normal in a cell, 2 complementary RNA strands is not, and the cell has special machinery that will destroy double-stranded RNA. By doing this, the RNA copies of the gene will never make it to the cell machinery that makes proteins, and the incorrect copy of the gene will not be made into incorrect protein. While this is great, because it avoids many of the problems of incorrect DNA insertion, since there is no insertion occurring, it has several limitations. Firstly, it is actually very hard to deliver the siRNA to the necessary cells. There are proteins in your blood called nucleases that destroy RNA and DNA in the blood. Furthermore, RNA cannot easily cross the fatty layer around our cells, called the plasma membrane. A lipid package, essentially made out of fats, can be used to package the RNA and deliver it to cells, although this can result in a strong immune response in some cases. In addition, unlike DNA-targeted gene therapies, RNAi medication would continually have to be taken, as in it's not a one-time dose, as the incorrect RNA copies of the gene are continuously being produced by the cell. How could we get around the problems that we've mentioned? CRISPR-Cas9 is an advanced genome editing tool that could solve many of these problems. CRISPR consists of two main components, the Cas9 protein, which cuts the DNA, and the guiding sequence strand, which has a complementary sequence to the gene of interest. You can think of the guiding sequence strand as stencils drawn across the paper to direct the artist throughout the sketch, and the Cas9 as scissors that cut the sketch once it's complete. The guiding sequence strand matches a specific sequence within our DNA that we would like to modify. The guiding sequence is embedded within the CRISPR-Cas9 system and searches for this DNA sequence to find its match. Once the match is found, Cas9 will then cut at that specific position. After that, CRISPR-Cas9 has the ability to edit in one of the following ways, depending on the situation. 1. It can cut something out and glue the rest together. 2. It can add a single one-letter change. Or 3. It can insert an entirely new sequence. Sounds like a great tool, right? It's precise, fast, versatile, cheap, and easy to use. You may be wondering by this point why it's not widely applied in clinical settings yet. Although CRISPR-Cas9 is very precise, it still has some off-target effects that could be harmful to us and that are still being more closely investigated in the lab. CRISPR-Cas systems are very powerful genome editing tools, and as a result, there are several ethical concerns with its use. It is important to note that CRISPR-Cas9 is now only used to edit somatic and stem cells to treat genetic diseases, and not to enhance or change other traits like your eye color, for instance, or making it taller. It also should not be used to edit germline cells, like egg and sperm, at this time. Let us explain why. Somatic cells are all cells in your body that are not sperm or egg cells. For example, these are your blood cells, heart cells, kidney cells, and so on. It's important to note that these cells are not passed on to your children, so any changes made there are not inherited by future generations. Changes in germline cells, however, do get passed on to the next generation, because if you remember, all cells in your body derive from the union of your parents' egg and sperm. Thus, any changes in those cells will be passed on to the individual and their children too. Since CRISPR editing still has some off-target effects and potential side effects in the long run are still unknown, germ cells are not edited at this time. Another big limitation is that the gene therapies we have outlined above are designed only for monogenic disorders, which are conditions that involve only one gene. Polygenic disorders, on the other hand, are diseases that involve multiple genes. You can consider in this diagram here that each circle represents one gene, and so polygenic disorders arise from a complex interaction of these multiple genes. Examples of such diseases include diabetes and coronary heart disease, and these cannot be fixed with the technology that we have now. Much more work needs to be done to understand how different genes play a role in these diseases, as well as adapting existing gene therapies to address the challenge of multiple genes. So this remains a limitation for the time being and a goal of the future. Gene therapy is still a very much developing field. As you've seen in this webinar, there are still several issues that have to be addressed, such as making targeting more specific and avoiding immune side effects. Despite this, the field has made massive leaps in the past 3 decades, and with CRISPR, we are currently standing at the cusp of a revolution in gene therapy. We are now much closer to one day being able to cure those with hereditary disorders, as well as treating somatic genetic diseases such as cancer. The future of gene therapy is bright. If you would like to check your understanding, please stay tuned for some questions. Question number 1. If a person has a blood-based disease, which method is most likely to be used? Question number 2. Which gene therapy would have longer-lasting effects, RNAi or viral gene therapy? Question number 3. Why are genetic diseases so difficult to treat? And question number 4. If you recall, the CRISPR system is a very powerful tool. Why can't we use it to edit germ cells to avoid heritable genetic diseases entirely within a family? We encourage you to pause your video at this time, answer the questions, and then resume watching to view the answers. The answers to the question are as follows. Question 1. If a person had a blood-based disease, you would most likely want to use an ex vivo gene therapy. So one of the limitations of ex vivo gene therapy is that cells must be easily accessible, and this is definitely the case with blood-based diseases, and so it is most frequently used with such disorders. Question number 2. Which gene therapy would have longer lasting effects, RNAi or viral gene therapy? The answer is viral gene therapy. If you recall, viral gene therapy involves insertion of DNA into the human genome, which is a permanent alteration. RNAi, on the other hand, involves giving a patient doses of RNA that intercept the mutated RNA of the cell, preventing it from becoming a protein. Because RNA is produced and destroyed by the cell all the time, a patient would have to continuously receive RNAi doses to maintain the effect. Question 3. Why are genetic diseases so difficult to treat? And so far, the prevailing issues have been adverse immune reactions and issues with specificity. And finally, number 4, why can't we use CRISPR to edit germ cells to avoid heritable genetic diseases entirely within a family? This is because it might affect the development of a fetus in unexpected ways or have long-term side effects that are not yet known. Thank you for watching our webinar, and we hope that it's been informative and that you've learned a lot about gene therapy.